We have been tasked with sequencing a purified protein to discover its primary structure. The primary structure determines its protein's structure and its function. The first step in sequencing our mystery protein is to degrade the entire chain using amide hydrolysis. This breaks apart every peptide bond in our chain and gives a mixture of free amino acids. Then, using an amino acid analyzer, we can determine which peptides are present and in what relative quantity. Utilizing a pH gradient, we can manipulate the charge on the amino acids to control the order in which they elute based on how their overall charge interacts with a negatively charged ion exchanged resin. Amino acids will elute in the order of lowest pKa to highest pKa value. After doing amino acid analysis on our unknown protein, we acquired a chromatogram with the following data. Comparing our results to a standard chromatogram, we can see that we have the amino acids threonine, serine, glutamic acid, cysteine, methionine, isoleucine, tyrosine, histidine, and arginine. Since each amino acid shows equal peak heights, we can see that each amino acid is present in equal quantities. Now that we know the amino acid composition of our protein, we need to determine the order in which the individual amino acids are linked. Because we have a small protein, we can use the Edman Degradation procedure to determine our amino acid sequence. To note, if we were dealing with a larger protein, approximately 50 residues or more, then we would need to break the peptide up into smaller sequences using enzyme hydrolysis.
The Edman degradation utilizes a phenylisothiocyanate reagent, which reacts with the free amino group on the N-terminus of our peptide. The first step in this reaction involves nucleophilic attack of the lone pair electrons on the nitrogen to the electrophilic C, which pushes these pi electrons onto the sulfur. We are then left with this intermediate. At this intermediate, a proton transfer occurs, and this nitrogen steals a hydrogen from this nitrogen, which involves a cascade of electron movement. Once at this intermediate, the carbonyl oxygen picks up an acidic hydrogen, which activates the electrophilic carbon and makes it susceptible to nucleophilic attack from the pi electrons of the carbon-sulfol double bond. This induces cyclicization of the molecule. Once that occurs, a water molecule comes in to steal the hydrogen, and the tetrahedral intermediate collapses to reform the carbonyl and expels the remainder of the peptide chain.
We are now left with the thiazolinone group, and the rest of the peptide is expelled.
The thiazolinone group then undergoes a rearrangement. It is first protonated by an acid at the nitrogen, which activates the electrophilic character of the carbonyl carbon. A water molecule then comes in and acts as a nucleophile, attacking the carbonyl carbon, which pushes the electrons up onto the oxygen. We then have a tetrahedral intermediate, which collapses back down to reform the stable carbonyl, and with that breaks open the cyclic structure by breaking the carbon-sulfur bond, which has a cascade of electron flow. We then have another carbonyl carbon, which is protonated by an acid. The lone pair on the nitrogen then act as a nucleophile to attack the activated carbonyl carbon electrophile. This reforms a cyclic structure, and a proton transfer occurs where the lone pair on the oxygen take a hydrogen from the nitrogen, giving these electrons the positive charge. We then have another tetrahedral intermediate, which expels a water molecule, and the lone pair on the oxygen drop back down to reform the stable carbonyl. A water molecule then comes in to steal the hydrogen to give electrons that positive charge, and we are left with the phenolethiohydrantione product.
The Edman degradation pulls a single amino acid off the N-terminus at a time. This allows us to figure out the order. Our first one was cysteine, followed by histine, and after we had repeated this 7 more times, we found the sequence of our protein.